Hello everyone, my name is Marina Lazic and I'm coming from University of Alberta in Edmonton. Today I will tell you the story about bioconversion of methane to biodegradable plastic, and I hope you will all enjoy. At the beginning, I would like to emphasize that biodegradable plastic is extremely eco-friendly material. Furthermore, with usage of biodegradable plastic, we will be able to reduce carbon emission, and this material is completely recyclable. Now, to understand the significance of biodegradable plastic, we first need to understand the significance of traditional plastic in our everyday life. And I'm sure that at one point we all use plastic bags and plastic bottles. Also, there are many medical devices made of traditional plastic, and who can actually imagine packaging without plastic? However, we are trying to stop using this traditional plastic and to use biodegradable one. And to understand why this matters so much, we first need to know what is traditional plastic made of. So one of molecules that is used for production of traditional plastic is called PET. PET is mixed with some additives which are creating a toxic material. Now, I'm sure we all know that PET-based plastic should end up in this blue bin. However, sometimes mistake happens and this PET plastic ends up in landfill container. In landfill, as we are piling up our waste along with the PET-based plastic, all this waste will be exposed to the environmental factors, which will create a toxic material that is called leachate. Leachate will leak into the soil and the ground and the water and will definitely threaten to harm our wildlife. And this is not what we want to happen. We want to prevent this. I believe it's clear that we need to find solution just to avoid this pollution. And solution is replacing a PET with polyhydroxybutyrate or PHB. PHB is precursor for production of this biodegradable plastic. And I'm sure if we succeed to replace PET with PHB, we will end up with a happy and healthy wildlife, which is required for happy and healthy planet, and that is what we all really want. Now, the question that might arise at this moment is, so if this PHB is so good, then why we didn't exploit this amazing material? What are we waiting for? Well, the problem is that currently PHB is mostly made from sugars, and sugars are very costly. Furthermore, using of sugars for PHB production will raise up food versus fuel debate. And now there is a question, what to do? How to ensure that we have made a lot of PHB that we can use for environmentally friendly material. And we are here offering solution strategy, which are bacteria that we know as methanotrophs. These amazing organisms are able to convert greenhouse gas methane during their active bacterial metabolism into the PHB. And methane metabolism is very well known. Methane is converted to methanol via methane monooxygenase, methanol to formaldehyde via methanol dehydrogenase, where formaldehyde is further converted to carbon dioxide in the set of dehydrogenases. The branching point that distinguishes two groups of methanotrophs is the way how they assimilate formaldehyde. While gamma methanotrophs are doing it through ramp pathway, alpha methanotrophs are assimilating formaldehyde through serine pathway. And for this talk, I will be focused on serine pathway because the organism, PHB producer, Methylocystis species Rockwell, belongs to alpha methanotrophs. Now, the first step in this project was to optimize bacterial biomass production. And there are two very important factors that are affecting bacterial biomass. One of them is carbon source, and the other one is nitrogen source. This bacterium can use either methane or methanol as a carbon source, and it can use ammonium or nitrate mineral salt as a nitrogen source. And we actually designed 4 growing conditions, which are here all color-coded using 2 carbon and 2 nitrogen sources. And we grow bacteria under each of these conditions, and we analyzed biomass production. And these are results that we achieved. So in this figure, the x-axis is showing the time of biomass analysis, where y-axis is showing optical density, which is indirect representation of bacterial biomass production. And as we can see here, the best growing conditions for bacterial biomass are methane, ammonium. Next logical step was to analyze pHb level under each of these 4 conditions. And we applied GCFID analysis, and these are results that we achieved. So the y-axis here is showing the percentage of pHb per dry weight under each of these 4 conditions. And as it's shown, the growth conditions that are promoting pHb production are methanol, ammonium. Notice, not the same like a biomass promoting conditions. So what we want to get is a lot of bacterial biomass, and we want that biomass mostly to be pHb. But that's not what we achieved. That is why we moved to the step 3, and we decided to analyze global metabolomic profile under each of these 4 conditions. And here is why. So pHb is considered to be industrially important or value-added compound. But pHb is not the only compound that can be considered value-added and industrially significant. There are others, and you will see later on why that matters. So, to analyze global metabolomic profile, we harvested bacterial biomass at defined time points, which are indicated by arrows, and we sent it to the profiling. And after detailed analysis, we successfully divided bacterial metabolism into the primary, which are methane, ammonium, and methane nitrate, and starvation metabolism, which are methanol, ammonium, and methanol nitrate. Primary metabolism is related to those metabolic pathways that are generating energy, where starvation metabolism includes those metabolic pathways that are stress-related. And here is the list of some metabolites produced under each of these 4 conditions, and some of them are industrially important, where the other ones are scientifically important. And I will start my analysis from scientific point of view. So when bacterium grows under stress-related or pHb-promoting conditions, methanol, ammonium, methanol will be converted into formaldehyde, and formaldehyde will be assimilated through serine cycle. What we see here is high accumulation of serine, which is indicator of stalled serine cycle under these conditions. Serine is accumulating while waiting reduction equivalents to come so the cycle can continue to operate. One of the most important products of serine cycle, acetyl coenzyme A, is going to be shunted to pHb cycle under methanol ammonium conditions. Under another stress-related conditions, methanol nitrate, we don't see accumulation of serine, but we see accumulation of another serine cycle amino acid glycine, which again indicates stalled cycle. Next, we see accumulation of beta-hydroxybutyrate, which is indicator of increased pHb cycle. And that's what we see, high pHb under these stress-related conditions. When methane is a carbon source and ammonium is nitrogen source, bacterium is growing perfectly well. And we don't see accumulation of serine because serine is constantly metabolized. And in this case, acetyl coenzyme A is not going to be shunted to pHb cycle. It will be shunted from pyruvate to TCA cycle, which we see through accumulation of TCA cycle derivatives, which are aspartate and glutamate. And that is why we don't see high PHB under these conditions. We have another growth promoting conditions, methane nitrate. And again, no accumulation of serine, fast metabolism. This time, we see other indicators of TCA cycle, which again tells us that this time acetyl coenzyme A is not shunt to PHB, it's shunt to TCA cycle. That is why we don't see high PHB under these growth promoting conditions. Now, we saw scientific point of view, but let's see what can happen from industrial point of view. So we know that PHB is commercially important, not just environmentally important. We know that the PHB precursor beta hydroxybutyrate is also important and produced under methanol, ammonium, or methanol nitrate conditions, respectively. But what we notice here that under biomass promoting conditions, there is accumulation of some other value-added compounds, such as amino acids, aspartate, and glutamate, or food additive succinate. So how can we use now this information? So for example, let's say we want to produce our environmentally friendly PHB in industry. We would say that we want to grow cells with methanol ammonium because these are PHB promoting conditions. The problem here is that these conditions are directing bacteria to the starvation, which results unfortunately into the low biomass. So if we want to industrialize PHB production, we would first have to grow bacteria with biomass promoting conditions such as methane ammonium, and then after achieving high biomass, we would initiate PHB production with stressing cells with methanol. However, under methane ammonium conditions, which are growth promoting conditions, we notice high production of succinate, food additive. So this is primary metabolism, a lot of biomass, which means if we want to industrialize succinic acid or succinate, we would simply grow bacteria under methane ammonium and get our product. But here is how we can make this win-win situation. Imagine that you have bioreactor fulfilled with our bacterium, and we are feeding this bacterium with methane. Bacterium is growing with methane ammonium, for example, or methane nitrate, producing high biomass. We can actually use this biomass to get succinate, because succinate is excreted into surrounded media. So we just collect supernatant, we get our succinate, and then we use the same biomass to treat it with methanol, which will lead to starvation and cause PHB production. So two value-added compounds, the same biomass. So our future directions here are to use this organism to create something called genome scale metabolic model, or GEM. So GEM is nothing else but simple integration of all available omics data, where metabolomics is probably the most important. So GEM is just detailed metabolic map that we already created. So what is actually metabolic map? So metabolic map is something very similar to the Google map. So in the same way how Google mapped the world, we are able to map bacterial cell. Think about that. It's pretty much the same. You have a methane inside the cell, and there are various roads that this methane can take to arrive target destination. And target destination is our PHB, which is bioplastic precursor. Once when we complete metabolic map, our goal is to develop genetic engineering strategy, which can result in now novel modified rock well, which will help us to convert large amounts of greenhouse gas methane into the large amounts of green products. That is how we will take care of our planet. Because remember, there is no planet B, and we all have to be aware of that. At the end, I would really like to thank to my both bosses, Dr. Stein and Dr. Sovajo, and to all lab research groups, friends, and family. And I would be happy to take any questions if you are interested to find out more. Thank you so much.